Hello, this is a lecture for second year students of medical faculty. Today, our topic is immune system, immune response, and allergy. Here you see the lecture schedule. We start with characteristic of immune system, the central and peripheral organs, and main cells of immune system, the lymphocytes. The second part is about immune response, primary and secondary, cellular and humoral, and special types of immune response like immunologic memory and immunologic tolerance. And third part is about allergy, their definition, their classification according to Coombs and Gell, and causes, manifestations, clinical mechanisms, and methods of diagnosis of four main types of allergies. Now we start with immune system. You know that we are surrounded with numerous um, bacteria, viruses, protozoa, fungi, toxins, cancerogens, mutagens, allergens, and our immune system protect us from these harmful organisms and substances. Immune system of human body consists of lymphoid organs and lymphatic cells. It provides identity and integrity of our organism. Immune system can distinguish own structures from genetically aligned, and it saves own structures and destroys foreign. Immune system consists of central and peripheral organs. Central organs are bone marrow and thymus. Before birth, we have the third central organ is embryonic liver. Peripheral organs are more numerous. It is spleen, pharyngeal tonsils, lymph nodes, lymphatic tissue of intestinal tract like pear patches and bronchi. The total weight of these lymphatic organs is approximately 1.5 to 2 kilos, similar to brain, for example. The central cell of immune system is a lymphocyte. Lymphocytes originate from bone marrow like all other blood cells, originate from stem cells, and then the development of lymphocytes maybe in two different ways. Some of them undergo the first step of their maturation in bone marrow, and these cells named B cells. Others transported to the thymus and undergo the first stage of maturation in thymus, they named T lymphocytes. After these cells migrate from central organs of immune system to peripheral, and in peripheral organs of immune system, they perform their protective functions. In peripheral organs, lymphocytes meet the antigen and it causes their multiplication and differentiation into effector cell. After T cell encounter antigen, it produces three types of effector cell: T killers or T cytotoxic, which can cause contact lysis of target cells. T helpers, it's a regulative cells, which activate um, the functions of T killers and B lymphocytes. T suppressors, they're also regulative cells, but their function is opposite to helpers. They stop immune response, they suppress activity of T killers and B cells. After B cell contact with antigen, it also multiply and differentiate into effector cell, and these effector cells named plasma cells. The function of plasma cell is production of antibodies. The function of T and B lymphocytes is different. T cells are responsible for cellular immunity, which is more effective against bacteria, protozoa, fungi, against intracellular bacterial and viral infections, and also tumor cells and foreign tissues. If foreign antigen is a large eukaryotic cell, the cellular immunity is more effective. B cells responsible for humoral immunity, which is more effective against extracellular infections. It's overwhelming majority of bacterial infections, and it's effective against viruses before virus invade human cell. And all antitoxic immunity is also only humoral. After B cell contact with antigen, it's multiply, differentiate into plasma cell. Plasma cells produce antibodies. Antibodies kill the antigen. T lymphocyte contact with target cell containing some antigen, activates, multiply, differentiate into effector cell, and effector cell kill this target cell with virus inside it. Other important cells of immune system is the macrophages. They participate in reactions of innate immunity and adaptive immunity. In immune response, their function is phagocytosis, antigen presentation, and activation of T cell. And here is a micrograph of macrophage with a pseudopodia to take this bacteria inside. And the natural killer cells, they're not the same as T killers. They can cause contact lysis of viral infected tumor or foreign cells. And this electronic micrograph, you see the target cell is a tumor cell, large cell at the center. And these small cells is the natural killers which attack the tumor cell and cause it lysis. Now we come to humoral immune response. A primary humoral immune response can very complicated mechanism with three types of cell involved in the cooperation. It's the macrophages, T helpers, and B lymphocytes. And what are the functions of each cell in this primary immune response? The first cells involved in the primary immune response, phagocytic cells and macrophages, they ingest foreign antigen, process it, and present in the surface for antigen-specific T and B lymphocytes. At the second step, T helpers involved, they contact with foreign antigen presented at the surface of macrophage. Macrophage release special substances, monokines, also named interleukins. They're important for interaction between leukocytes, interleukins. And the substances activate T helper for proliferation and differentiation into effector cell. And the substance is important also for activation of B cells and natural killers. While contact between T helper and macrophages, T helpers have the CD4 receptors and macrophages also named antigen presenting cells. You see the phenomenon of double recognition, very important in immunology. At the surface of macrophage, there is a foreign antigen processed inside and own antigens, major histocompatibility complex antigens. And the T helper recognize both types of this antigen at the same time. Only if it meets both types of antigen, it can be activated. You know that immune system can differentiate own antigens and foreign, and this property is at the level of cell T helpers. At the next step, activated T helper transfer the antigen to B cells and release lymphokines, interleukins, substances which necessary for activation of B cells. And it stimulates their division and the maturation into plasma cell. And plasma cells synthesize antibodies, its immunoglobulins classes G, A, and E. No immunoglobulin M. It synthesizes in a simpler way without T helpers and macrophages. Plasma cell produce antibodies, and antibodies neutralize antigen or cause it lysis or cause clumping, agglutination or precipitation or opsonization. So they kill the antigen in different way depending on class of this immunoglobulins. And here you see the schematic diagram summarizing the mechanism of primary immune response. Firstly, the macrophage involved in this process, it ingests foreign bacteria or virus processed antigen present. It is at its surface The next step is a contact with T helper, double recognition, release of interleukins, activation of T helper, transfer of this antigen to B cells, only for specific B cells. This cell have their own receptors to recognize the same antigen, and only specific B cells may be activated. Transfer of antigen, release of interleukins, activation of B cells, their multiplication, differentiation into plasma cell, and production of antibody is the final step of humoral immune response. After first contact with antigen, the plasma cell produced and cells of immunological memory. Plasma cells are very metabolically active and they live only some days and then die. But cells of immune memory stay alive many years. They live in lymph nodes, in tonsils, in organs, and they wait if the same antigen invade human body. In this case, memory cells react with them immediately and produce secondary immune response. And what is the main difference between primary and secondary immune response? It needn't involvement of macrophages and T helpers. In primary immune response, it is a cell cooperation. In secondary immune response, it needn't. So the production of antibodies is different in primary and secondary immune response. In primary immune response, the high titer of antibody can be detected no earlier than in 2 weeks after invading antigen. Mostly immunoglobulins E, M produced because they needed the participation of the helpers. And some days later, we can detect immunoglobulins G. And in secondary immune response, memory cells activated directly with antigen. They multiply, differentiate into plasma cell, and the production of antibodies is faster and in higher titer than in primary immune response. So in secondary immune response, the synthesis of antibodies is faster because no cell cooperation, and the titer of antibodies higher, much higher than in primary immune response, because it is some selection of specific lymphocytes. Due to somatic mutations, we have very different T and B cells in our body with receptors which can react with all known antigens and some unknown antigens. But for each antigen, we have only a few cells. If one antigen enters the human body first time, it activates only specific T and B cells. These cells start to multiply and produce the whole clone on memory cells. And in next invading, the antigen reacts with this whole clone. All these numerous cells start to multiply, turn into plasma cells, and produce much more immunoglobulins than in primary immune response. So it's recommended the multiple administration of vaccine to produce strong immunity. Vaccines like DTP or MMR injected 3 times to increase the titer of antibody and produce long-lasting and strong immunity. The same reason is for hyperimmunization of animals, which is important in production of immune serum. We inject the same antigen like tetanus toxoid in the horses, and the very high titer of antibodies accumulate in the serum of this animal, and we can take the serum and use like medication for people suffering from tetanus. And knowledge about differences in primary and secondary immune response is important in diagnosis of acute and chronic infection. If it is acute infection, it's a primary immune response, and we detect mostly immunoglobulins E, M. In chronic infection or in the period of recovery, the immunoglobulins G detected predominantly. And detection of these two types of immunoglobulins important in diagnosis of congenital infections like HIV, hepatitis B, toxoplasmosis. If we suspect this infection in newborn, we do blood test. And if we find only immunoglobulins G, it's a good result. It means that only immunoglobulins G were transported from mother's body through placenta, and it's no infection. But if we detect immunoglobulins M, it's a result of primary immune response in organism of newborn, and it means that this baby is infected and needing immediate treatment. Now we come to next type of immune response. It's cell-mediated immune response. Three types of cell involved in it. It's T killers, T helpers, and macrophages. Macrophages ingest antigen, process it, present in the surface, and release interleukins to activate T helpers. T helpers, after double recognition, activated, they multiply, release interferons, release interleukins to activate natural killer cells, and to activate T killers. T killers recognize the microbial antigen like this at the surface of target cell and produce special proteins, granzymes, and perforins, which cause the damage of cellular membrane, kill the target cell with pathogenic bacteria viruses inside it. And this pathogen is the same as that, because adaptive immunity, immune response, is always specific, directed against certain bacteria or virus. When T killer contact with target cell, the double recognition take place. At the same time, T killer recognize own antigens of major histocompatibility complex, the normal cellular antigens, and foreign antigens. It may be viral or tumor antigens. Only if two types of receptors connected, the T killer can kill this target cell. What is the function of T helpers in the cellular immune response? It's the recognition of antigen and stimulation of T killers. T helpers release interleukins, which is important for multiplication and differentiation of T killers. And T killers cause the lysis of target cell. The special type of immune response is the immune tolerance. It's a negative immune response, not absence of immune response. If immune response is absent, this condition named immunodeficit. But normally, immune system react, give positive immune response for foreign antigens and negative for own. It is active suppression or inactivation of some lymphocytes which can react with certain antigens. So normally, the tolerance must be against own antigens. And artificial immune tolerance is also important in clinic, especially for transplantation of organs and tissues. And now it's not problem to take any organ from donor and insert to the body of recipient. And this organ would perform their function for some days, but later immune system of recipient start to produce antibodies and T killers, and it leads to rejection of graft. And there are some methods to suppress activity of immune system to induce artificial tolerance, maybe high dose tolerance, low dose tolerance, or drug induced. But this very interesting questions is out of our lecture. And the next part of our lecture is devoted to allergies. Allergies also type of immune response. It's a reaction for some substances, allergen. What may be allergen? Any substances. Which organs may be affected as a result? Any organ. So allergy is very important condition. this knowledge very important for all doctors. Allergy, also named hypersensitivity. This term originated from Greek, and directly it means allos, with other, and ergon, work. So it's improper work of immune system. It reacts too much for some harmless substances. Actually, allergy is altered immune response following secondary contact with the same antigen. At the first contact, this hypersensitivity only induced, but no clinical symptoms. Clinical symptoms appear only in secondary and other contact with the same allergen. Allergy is a consequence of organism sensitization. It's some changes in the immune system, it's production of antibodies or T cells. And in general, allergy is a condition in which invading of antigen causes immune response that harms the body. So for allergy, immune response more harmful than antigen. Substances which cause allergy, named allergens, they have property of complete antigen or incomplete or hapten. The origin of allergens very different. Some of them infections, the part of fungal cell or bacterial cell or viral particles. And there are a lot of non-infection antigens, maybe drug allergens, food allergens. The plant pollens is powerful antigen, and there is antigens of animal origin. The ticks or mites, dusts also contain allergens. And here you see the classification of allergies according to Coombs and Gell. This classification is based on the mechanism. Allergy is an immune response. Immune response may be cellular and humoral, and allergy may be mediated with antibodies or effector T cells. And there are at least the 5 types of immunoglobulins in human body. And first type of allergy associated with immunoglobulins E, and it's named immediate reaction because it appears very fast in some minute after contact with allergen. The second type, mediated with immunoglobulins G and M, and it directed against allergens which are type with a part of cellular membrane. It causes cell lysis, and this reaction named cytotoxic. Subtype also associated with immunoglobulins M and G, and in it the immune complex antigen antibody are produced. So this reaction named immune complex reaction. And fourth type of allergy associated only with T cells, and it named cell-mediated or delayed immunity allergy. And here you see the mechanism of immediate hypersensitivity or anaphylactic reaction. This process starts with invading of antigen. Antigen react with B cells. B cells multiply, differentiate into plasma cell. Plasma cells produce immunoglobulins E. Immunoglobulins E attach to the surface of mast cell. In this state, they circulate in bloodstream. And here you see changes in immune system, but no clinical appearance. Clinical symptoms appear after the same antigen invade human body next time. It binds with immunoglobulins E at the surface of mast cell, and it causes the release of histamine and heparin from mast cells. And all the symptoms occur. The sharp contraction of smooth muscles, increase of vascular permeability. Due to these processes, blood accumulates in the periphery and the blood pressure fall down. Other symptoms, it's nausea, diarrhea, maybe bronchospasm, laryngeal edema, and the severe case, even shock and death. When it's possible, it may be after injection of some medicines, like foreign serum, used in treatment of tetanus, gas gangrene, botulism. It's a powerful allergen and can cause death of patient in some minutes. How to avoid this complication? It's necessary to do the skin test before injection of suspected allergen. Other condition which have the same mechanism of immediate hypersensitivity is atopy. Atopy is a strange reactions for completely harmless substances like different types of food. Some people may have allergy for orange, other for egg yolk, for sea products, peanuts. Others have this abnormal reaction for land pollens, for animal hairs, house dust, and other. It's hereditary form of allergy, and it's only in human, not in animal. The mechanism of atopy is very similar to anaphylactic shock associated with elevated level of immunoglobulin E. It's necessary the first contact with allergen, activation of B cells, they turn into plasma cell, production of immunoglobulins E. Immunoglobulins E attached to the mast cells. This is the first step of process, changes in immune system, but no symptoms. Then next, contact with same allergen. It reacts with specific immunoglobulins E at the surface of mast cell. Mast cells release histamine and heparin and damage some organs, causing conjunctivitis, runny nose, skin rash, and other symptoms. And diseases with this mechanism is bronchial asthma, hay fever, urticaria, and some others. They are widely spread in human population. And in this table, you see the summarized information about clinical appearance of most important reactions of immediate hypersensitivity. It's very important to diagnose which allergen causes this condition in human. We can perform skin tests for this purpose, injecting a small amount of allergen intradermally, and in some minutes we see result. If we see a red spot, the reaction is positive, so the patient is have hypersensitivity for this substance. Is no red spot, no allergy. Other method is serologic test, when we take blood and do precipitation test with suspected substances to know which of them causes allergy. And then it's enough to avoid the contact with the substance to prevent appearance of clinical symptoms. Second type of allergic reaction is cytotoxic reactions. Immunoglobulins G and M participate in. They bind with allergen, which is a part of cellular membrane, and if antigen antibody present in bloodstream, it induces the activation of complement system, and the result is fast phagocytosis of this cell or the lysis due to membrane attacking complex producing this complement. And look at the scheme, two types of immunoglobulins involved. As a component of immune system involved in this process, it's complement system. Some active subfractions attract other cells which participate in inflammatory process, or complement can cause the lysis of this target cell. Mostly it's a cell of our body. And which diseases have this mechanism? It's a drug allergies. When some medicines attach to cells of human body, like phenacetin can be attached to erythrocytes. In this case, these small molecules turn into complete antigen. They stimulate immune system to produce immunoglobulin E, M, and immunoglobulin G against these cells. And as a result, complement system activates and causes the lysis of this cell, own cell, but with foreign antigen. As an example is hemotransfusion shock, also antigens, erythrocytic antigens, and antibodies, isohemagglutinin present in it. If incompatible blood group is injected into recipient, it can cause this condition. It's lysis of cells due to participation of immunoglobulins and complement system, so it's allergic reaction. And the hemolytic disease of newborns, the same as rhesus conflict, also an example of cytotoxic hypersensitivity. Involved components, it's immunoglobulins G of rhesus negative mother and erythrocytic antigens, rhesus factor, positive rhesus factor in erythrocytes of fetus. And the result of this allergic reaction is miscarriage and other severe complications in newborns. The third type of allergic reaction is immune complex. Small antigens, small soluble antigens, and antibodies classes M and G participate in it. They produce immune complexes. They're too small to be phagocyte. And there's a accumulation of these immune complexes in the blood vessels, in kidneys, in skin, in lymph nodes. And if immune complex antigen antibody present, it causes activation of complement system, and active subfractions attract neutrophils and basophils. Neutrophils produce proteolytic enzymes, basophils release histamine and heparin, and it causes inflammation and necrosis of endothelial cells in blood vessels, and attachment of platelets, and thrombosis, necrosis as a result. And which diseases have this mechanism? It is a serum sickness, which develop after injection of foreign serum, like antitoxic diphtheria serum for treatment of diphtheria. The immunoglobulins G present in the serum is an antigen for human body, and the immune system produces own antibodies against these foreign immunoglobulins. And these immune complexes deposit in blood vessels, in kidneys, in skin, and activate complement system and cause inflammatory process. It is not life-threatening disease because we inject foreign immune serum only one time. The most severe case is autoallergies like systemic lupus erythematosus. In some people, immune system start to produce antibodies against own DNA. And these immune complexes deposit in skin, causing these red spots, and in kidneys. And eventually, the disease leads to renal failure and death. And it's incurable. We can only slow down it, administering anti-inflammatory drugs, but we cannot stop this process. And other interesting disease is rheumatoid arthritis. In some people, immune system produces immunoglobulins M against own immunoglobulins G. These immune complexes deposit in joints, and the sequences the same: activation of complement system and chronic inflammatory process. And the fourth type of allergy is the cell-mediated or delayed hypersensitivity. It caused only with T helpers, T cells, and sometimes macrophages involved in it. T cell recognize target cell, multiply, produce interleukins to attract neutrophils, and result is inflammatory process, local inflammatory reaction. Often this process develop as infection allergy, especially when bacteria multiply inside cells, like in gonorrhea, in chlamydiosis, tuberculosis, brucellosis, and others. In this case, the immunity mostly cellular, and the allergy also cellular. And what's important, this reaction is specific, so we can use it for diagnosis. It's an intradermal injection of allergen, like Mantoux test used in diagnosis of tuberculosis. It's very specific because T cell can react only with that antigen, which causes their appearance. After injection of microbial allergen, reaction developed in 8 to 72 hours, so it named delayed hypersensitivity. And if reaction is positive, it means that live pathogen present in organism. In Mantoux test, this reaction positive in infected people and in people vaccinated with BCG vaccine. And the similar test also used for diagnosis of chronic gonorrhea, chronic chlamydiosis, brucellosis, anthrax, and some other diseases. In that cases, reaction positive in ill people, in infected people. And the other diseases with the same mechanism of delayed hypersensitivity is the contact dermatitis, which can be maybe caused with poison plants like poison ivy, heavy metals, and disinfectants and medications. These substances have a property of hapten. After first contact, they bind with skin proteins, they turn into complete antigen, they activate immune system to produce effector T cells. And second contact causes these symptoms: redness, swelling, wet lesions on skin. It's a pity, but this disease is often diagnosed in medical professionals due to permanent contact with disinfectant and some medications. And how to cope with this problem? It's very important to know which substance causes disease and avoid contact with it. The effector T cells have a short time of life, some day, and if not next contacts, no activation of immune system, the symptoms eventually cease even without treatment. And this table summarizes information about all 4 types of allergies. Here you see the mechanism, description, and the diagrams, and the clinical examples about each of them. And that's all I want to tell you today. And if you have any questions, write for our email address, the same as for you written works, new papers. And thank you for your attention.